The aim of this procedure is to label chromosomes with paint probes to identify chromosome aberrations and segregation defects in mice and humans with polycystic kidney disease. This is accomplished by first harvesting cells from humans or mice and treating with colsamid, followed by fixing and preparing metaphase spreads on glass slides. Next, the slide is pre-treated to remove extra RNA and cytoplasm which might interfere with the hybridization. This is done by digestion with RNase and pepsin, respectively. Then, chromosome and probe denaturation is performed, followed by hybridization to label chromosomes. The final step is detection of the probe with fluorescent dyes. Ultimately, results can be obtained that show chromosome numeral and structural abnormalities through fluorescence microscopy and image capturing and analysis by Skyview software.
The main advantage of this technique over existing methods like classical Giemsa bending techniques is that it provides additional information about complex chromosomal aberrations like chromosomal translocations and complex chromosomal rearrangements that cannot be characterized by classical bending techniques.
This method can help answer key questions in the polycystic kidney disease field, such as the mechanism of cystic kidney formation. Demonstrating the procedure will be Dr. Wissam AbouAlaiwi, a talented senior postdoc from our laboratory.
To begin, grow mouse endothelial cells in DMEM containing 10 to 15% fetal bovine serum and 1% penicillin streptomycin at 37 degrees Celsius and 5% carbon dioxide until the cells reach 70 to 80%.